Hello, everyone, and welcome. Today, we're diving deep into one of the most incredible processes in all of biology, spermatogenesis. You can think of it as a super sophisticated, microscopic assembly line. Our goal today is to walk through every single step from the very first cell all the way to the final product. So, to kick things off, let's start with a number. 300 million. Just let that sink in. That's roughly how many sperm cells a healthy male produces every single day. It's a staggering number, and it really speaks to the efficiency of this biological factory. And that, of course, leads to the big question. How on earth does the body manage this? How does it pull off this amazing microscopic feat of engineering, day in and day out? Well, that's exactly what we're going to unpack together. Here's our game plan. We'll tackle this in 6 parts. First, we'll check out the location, the factory itself. Then, we'll meet the key cells involved. After that, we'll get into the 2 main production phases, look at how the whole thing is controlled, and then wrap it all up with a nice summary. All right, let's jump right into section 1, the sperm factory. To really get this process, you first have to know the lay of the land. Now, before puberty, the germ cells, these are the very early precursor cells, are basically just sitting there dormant inside these solid structures in the testes called the sex cords. But then, puberty hits, and that's when a massive change happens. These solid cords actually hollow out, creating a central channel, or a lumen. And once that happens, they get a new name, the seminiferous tubules. This right here, this is our factory floor. It's the exact spot where all the magic of spermatogenesis happens. Okay, so we know the where. Now let's meet the who. We've got a few key players in this process, and knowing what each one does is absolutely crucial to understanding the whole story. First up, we have the Sertoli cells. Honestly, you can just think of these as the nurse cells of the testes. They are the ultimate support crew. They provide physical structure, they bring in nutrients, they take out the waste, and they even shield the developing sperm from the guy's own immune system. Seriously, without Sertoli cells, this whole process would just fall apart. And then we have our main character, the germ cell, as it goes through its different life stages. You can see it's not just one big change, but a whole series of steps. It all begins with the spermatogonial stem cell. That gives rise to type A and B spermatogonia, which then become primary and secondary spermatocytes, and finally, spermatocytes. We're going to look at each of these in just a bit. So, that brings us to the first major phase of production, division and multiplication. The name of the game here is just numbers, plain and simple. We need to take a small group of stem cells and turn them into a massive army of precursor cells. It all kicks off with those spermatogonial stem cells. Every so often, they divide. One of the new cells stays behind as a stem cell. Got to keep the supply going, right? While the other one commits to the journey and becomes a type A spermatogonium. These type A cells then go through a bunch of rounds of mitosis, which is just regular cell division, to make a whole pool of identical copies. The very last of these divisions makes type B spermatogonia, and when they divide, we get our next major player, the primary spermatocytes. And right here, we've hit a huge turning point. You see, up until now, every division has been mitosis. That means we've just been making identical clones with the full 46 chromosomes. But a sperm cell needs to have half that amount, right? Only 23. This is where we switch gears to meiosis. The primary spermatocytes are about to go through a special kind of division that's going to cut that chromosome number in half. So here's how it works. Each primary spermatocyte goes through meiosis 1 and splits into 2 secondary spermatocytes. These guys are now officially haploid. Then, really quickly, those 2 cells go through meiosis 2, which looks a lot like mitosis. And that gives us a grand total of 4 cells. And we call these cells spermatids. They're the final output of the meiotic phase. Now here's something that is just fascinating. When these cells divide, they don't completely separate. They stay connected by these little channels called cytoplasmic bridges. And this is so important because it forces the entire group of cells that came from that one original spermatogonium to develop at the exact same time, all perfectly in sync. It's an amazing bit of biological engineering. And that brings us to the second major phase, which is called spermiogenesis. Now, quick point of clarification, don't mix up spermiogenesis with spermatogenesis. Spermatogenesis is the whole shebang. Spermiogenesis is just this final, incredible transformation step. We've got the right number of cells with the right number of chromosomes. Now it's time to build the actual sperm. And what a transformation it is. The spermatid starts out as this simple round cell, and it gets a complete makeover. First, it forms the acrosome. That's a cap full of enzymes it'll need to penetrate the egg. Second, the nucleus gets super condensed, packing all that DNA into a tiny protected space. Third, it builds the neck, the middle piece, and of course, the tail, which is the engine for movement. And last, it sheds almost all of its cytoplasm to become sleek and streamlined. And would you look at that, the final product? You can see all the parts so clearly. The head with the DNA and the acrosome cap, the midpiece, which is just jam-packed with mitochondria to power the tail, and the tail itself, the flagellum that makes it swim. It's a perfect case of a cell's structure being perfectly designed for its job. Now, a process this complicated obviously needs some very precise controls. So what's running the show? What are the master signals that regulate spermatogenesis? And, how long does this whole journey actually take? The whole thing is run by hormones. It starts up in the brain with the pituitary gland, which releases two key hormones. First is luteinizing hormone, or LH. LH tells the Leydig cells, which are located between the seminiferous tubules, to make testosterone. Second is follicle-stimulating hormone, or FSH. FSH talks directly to our friends, the Sertoli cells, and you need both testosterone and FSH for those Sertoli cells to do their job of supporting sperm development. So, what about the timeline? From the moment a spermatogonium starts its journey until a fully formed spermatozoa is ready, the whole process takes about 74 days. Yeah, over 2 months. And what's really wild is that a huge chunk of that 74-day trip is spent in just one single phase. Primary spermatocytes hang out in the prophase of meiosis I for about 22 days. That long delay is absolutely critical for something called crossing over, where the chromosomes swap genetic info. This is the step that shuffles the genetic deck and creates diversity. All right, we've covered a lot of ground. Let's pull everything together into a nice, high-yield summary. This table is basically your cheat sheet for the whole process. Just look at the key transitions. We start diploid with the spermatogonium, which multiplies by mitosis. Then the primary spermatocyte, still diploid, is the one that kicks off meiosis. The big change to haploid happens right after meiosis I, giving us those secondary spermatocytes. And finally, the haploid spermatids go through that big transformation, spermiogenesis, to become spermatozoa. This is perfect for studying. But we have one last crucial point. At the end of spermatogenesis, even though the spermatozoa look the part, they aren't fully functional yet. They can't really swim properly. So from the testes, they get moved into this long coiled tube called the epididymis to finish their training, so to speak. I like to think of it like this. The seminiferous tubules build the car, but it's in the epididymis that the car gets its keys and the driver learns how to use the gas pedal. This final maturation step, where the sperm gain their motility and the ability to fertilize an egg, is the essential final chapter in the story of spermatogenesis. And understanding this whole journey is just fundamental to understanding male fertility.